Hi, welcome to my poster. My name is Sam Hamilton. I'm a graduate student at Northwestern University, Chicago, and I'm here to briefly overview the information in my poster entitled Machine Learning Informed Guidelines for Optimal RNA-seq Quality Control. So RNA-seq has become a ubiquitous tool in biology research. However, something that is lagged behind the broad use of RNA-seq is the establishment of how to identify poor quality RNA-seq samples. While there's no shortage of metrics that can be used to quantify technical aspects of RNA-seq quality assays, there's been little research into which of these metrics is the most informative of sample quality. In addition, tools that support RNA-seq QC, such as FastQC, are usually focused on a small slice of this protocol, which means holistic analysis of RNA-seq samples. So to address this issue, supporting holistic quality control analysis, our lab has developed this tool called Quality Control Solver for Diagnosing RNA-seq, or QC Doctor. QC Doctor takes as input a table of data with different information on RNA-seq quality for a data set, and then produces two primary outputs, a set of sample reports and a summary heat map. To go into the summary sample reports, you can see that for a collection of different metrics, which together broadly describe the RNA-seq protocol, individual samples value is marked in teal and placed into context with the distribution of values from the rest of the data set. A value falls below a particular statistical cutoff can be flagged. QC Doctor will flag it with either a fail or a warn. QC Doctor is designed to be customizable, and the background distribution and the fail and warn cutoffs can all be customized to an experimenter's individual needs. This information is then summarized in a summary heat map, which allows the researcher at a glance to identify which samples are being flagged the most, potentially indicating their poor quality for further investigation. So to test QC Doctor, we applied it to a data set, 252 alveolar macrophage samples collected as part of the successful clinical response pneumonia therapy project, shorthand script. And what we found was that it was able to successfully identify poor quality samples, but also poor quality samples suffering from a variety of different issues. So to just give some short examples, so for B1101, we found that this sample had a large amount of reads trimmed, and in addition, that even after trimming, it had a high amount of adapter content, suggesting the sample suffered from adapter contamination. So, having done this, our next goal was then to establish which quality control metrics were best for identifying poor quality RNA-seq samples. So to do this, we established three endpoint metrics of quality, which each captured a different aspect of sample quality. The first being sample correlation, captured how aberrant a sample's expression was. Then we did unique genomic positions, which captured to what degree a sample had been sufficiently sequenced. And then finally, cell type score, which captured how much a sample expressed genes typical of alveolar macrophages. So looking at the relationship between these endpoint metrics and a panel of QC metrics, interestingly, we found that experimental quality control metrics, which are those taken before sequencing to determine whether a sample is suitable for sequencing, were not that closely associated with sample quality. So you can see here that while many poor quality samples have a low RNA concentration, not all samples that had a low RNA concentration ended up being poor quality samples. In fact, most ended up being good quality samples. Overall, this suggests that many samples that might be being discarded because of low experimental quality scores could actually turn into good libraries where they sequenced. Next, we wanted to directly answer our fundamental question, which was what quality control metrics were the best predictors of sample quality by fitting a machine learning model predicting quality in the script data set. So to do this, first we binarize the data set into good and bad samples by setting a specific cutoff, and then calling all samples that had scores below that cutoff for two of the three endpoint metrics as poor quality samples. Then using this binarized data set, we then tried to predict whether the sample was good or bad quality just using the QC predictors. And so we found that we were able to predict what samples were good or poor with very, very good accuracy, with an area under the receiver operating curve of around 0.93. We then tested this on an external data set, a lung transplant set of 52 alveolar macrophage samples from a lung transplant data set, and received a similar high accuracy of 0.9, indicating our model was not overfit to the specificities of the script data set. We then wanted to look at what QC metrics were the most important predictors of sample quality. overall, we found the most important predictor was the number of detected genes, with a particular fall off in the sample quality around 10,000 detected genes. Overall, this suggests that number of detected genes is an underused metric for determining sample quality in RNA-seq samples. Last, we wanted to go a step further and look if there are any specific interactions that were particularly affecting the model. And so we developed this tool, this model agnostic tool for quantifying variable interactions in machine learning models. And then using it, applying it to our model, we found that there was this interaction in the percent of duplicate reads and then the percent aligned to multiple loci, finding that samples that had a high percentage of duplicate reads, but also poor alignment, were more likely to be poor quality samples than either of those metrics independently would suggest. This is intuitive because if you have a sample that has a lot of duplicates but also poor alignment, it probably means you have a library complexity issue. Overall, in this work, we've just presented QC Doctor, which is a tool for holistic RNA-seq quality control. We presented data describing the best features for assessing RNA-seq quality. And then we've also described this model agnostic tool for quantifying variable interaction in machine learning models called ALPlot Plus, and it's all available at github.com/hamhamilton. So if you want to see me talk about this in person, I'll be at the conference. Looking forward to seeing you all there. Take care. Goodbye.